Today we're going to be continuing on with the alternative practical past paper. So just a very quick recap of what we looked at last time. We had an experiment looking at the effect of temperature on the water uptake in celery stalks. So we had warm stain and cool stain, and we had celery in, and we went through it, and the stain comes up the celery stalk. So we looked at various different questions, looked at results, and looked at rate of movement, and we discovered that in a warmer stain, then the rate of movement is higher. So they asked us to consider the errors of the experiment, and then we moved into this question where we stopped. So this question now says that some farmers wanted to investigate the effect of humidity on the rate of transpiration in celery stalks. So the last one was the effect of temperature. Now we're looking at the effect of humidity. So we've changed things a little bit. Figure 1.5 shows celery plants being harvested. One method of measuring the rate of transpiration is to record how long it takes a red stain to travel up the xylem tissue in a celery stalk that still has leaves attached. So there's another slight difference. Plan an investigation to determine the effect of humidity on the rate of movement of water through leafy celery stalks. So in this original experiment that we worked with in this question, there were no leaves on those stalks. So we were just looking at movement. Now we're looking at movement as a result of transpiration as well. So we need that leaf on top as well. So remember, transpiration is that loss of water from the leaves. So as the water is lost from the leaves, then it's pulled up the stalk. So now we want to say, okay, so looking at that rate of transpiration, what's the effect of humidity? So we need to now plan an investigation. There's our usual collection of lines and our usual 6 marks. So we've got to think through what is it that we need to do. So if we're testing the effect of humidity, so the effect of humidity, that is our independent variable. And what do we want to do with the independent variable? We need lots of different levels. So in our temperature experiment, we just had warm and cool, but we want to be a little bit more thorough than that when it comes to this. So remember, we're aiming for 5 different things. So we're going to test at, okay, I'm actually going to put this into bullet points for today, just so that we can clearly see, because we haven't done many of these questions together already. Okay, so we're going to test at different humidities. So we're first dealing with that independent variable. But we don't want us to say different. That's far too vague. We want to have 5 different humidities. So we can have no humidity, 0%, 20%, 40%, there's 3, 60%, and 82%. I'd rather go to 0 than 100 on this one, because 100 is in the rain. It's going to have all sorts of other impacts on what we're doing. So let's go from 0 to 80. And humidity, we measure in percentage. Okay, so what are we going to control? Okay, if we want to test humidity, we have to control every other variable. So we're going to control temperature, of course, because we were reminded in the previous one that temperature was a factor. So temperature, wind speed, light intensity, carbon dioxide levels, and leaf surface area. These are some ideas of what you could control. So what are the ones that you would want to highlight in this response? Okay, you would want to highlight a few of these. Definitely want to highlight temperature, because temperature we've seen is a massive effect. Wind speed is also a big one to highlight here, because wind speed, of course, blows that moist air away from the leaves. So that is a big one. The other big one for humidity would be the leaf surface area, because we're dealing with, they told us, leafy celery stalks. So you wouldn't necessarily go and list all of these, okay, but it's the idea that we're going to control lots of different variables when it comes to testing one, when it comes to something like transpiration. But we want to be particularly sure that we're controlling those things that are going to affect the plant's perception of the humidity. So temperature, wind speed, and leaf surface area. Okay, so we're going to then, we've got our independent, we've got our controlled. What's our dependent? So our dependent is some measure of the movement of water. So you could use a stain, same like we did for the temperature, and then you would measure movement of a stain. So we're going to measure the distance moved. I'm getting myself back to front. Obviously, I'm having a classical Monday today. So we're going to measure the distance moved by the stain up the stem. And we know that 10 minutes works, so let's keep it at 10 minutes. So we're not going to go through that full description that they've given us already in the experiment that we worked with, of put it in the stain and then wait for 10 minutes, because that's like repetition. When we're planning these investigations where we're having a slight change, what we want to do is to focus on the things that are different. So yes, we need to, so things like controls and we're measuring the distance of the same roof, but what's important is testing the different humidities. What are we controlling when we're dealing with humidity? So repeating pieces of the method from the previous one isn't really going to get us marks. We want to obviously state that's how we're going to do it, but we don't want to go into lots and lots of details about how we're going to work with this thing. Okay, now they want us to actually investigate here the rate of water movement. So measuring the distance isn't actually enough. We have to then calculate the rate. So rate is distance over time in this case. So we would divide the distance moved by time taken to get rates to compare. So we're going now, we've got our independent, we've got setting up that sort of controlled environment, and now measuring our dependent, but we also got to convert that dependent into something that's actually going to answer the question. The question wasn't just compare distance moved, it was the rate of movement. So we need to tell them exactly how we're going to go from distance to a rate. What do we always do when we create a new investigation? We always have repetition. So we're going to repeat 5 times our magic number at each humidity level. So 5 times at 20%, 5 times at 40%. So we've got a lot of different experiments to run here. But remember, this is a theoretical situation. It's how would we run this experiment if we had like everything we needed and we could actually do it properly. So that's what we're aiming for. Repeating 5 times for each measurement of that independent variable. And the last one, of course, what we always do when we are doing any experiment is that we give them a safety precaution. It's like, what are we working with in here that's going to be potentially dangerous? Okay, and then how are we going to deal with that? So working with humidity, it's not too bad, but the stain, of course, is still here. So we would always want to recommend that whoever's doing this experiment works with gloves when they're working with the stain. So we're going to wear gloves when working with the stain. And you can see that we've got actually plenty of space here to turn these into nice sentences that then work through all of these different elements that we've got to consider when we are planning an investigation. Independent variables, control variables, measurement of the dependent variables, repetition, and safety. 5 important points, and if you cover each of those 5 points properly, then you're definitely going to make your 6-mark allocation. So that was the end. You can see that's the total of 19 marks for the end of our celery question. So any other points you guys want to query about the celery question before we move on to question 2? I've got to see another safety thing is wearing a lab coat. So yeah, lab coat would be another safety precaution. The thing is that when you're in lab, as soon as you go in the lab, you should have your lab coat on. So it's not so much of a specialized one. So what you want to focus on when you're pulling out those safety things is, okay, what's special about this experiment? Because in theory, we're already in lab coats already. Any other queries or comments for this question? Okay, cool. So then let us move on to question 2. And we see we've got something completely different. So figure 2.1 shows a photomicrograph of a bronchus surrounded by alveoli and other tissues in the lung. So if we look at it, we can see, okay, these little squiggly bits are our alveoli. The bronchus wall is that whole thick piece there. Then the bronchus lumen is the gap in the middle. So a lumen is always the piece in the middle. A part 1 says, make a large drawing of the bronchus shown in figure 2.1. Do not include any of the alveoli or other tissues in your drawing. Do not label your drawing. So what they're asking here is just that bronchus. Plain, simple, clear. What are the different things that are making up the walls of your bronchus? Okay, so we are drawing exactly what we are looking at, and we are following that instruction of do not label. Remember, if they want us to label something, they will tell us to label it. So now we have an interesting space issue. So let us see how small I can draw. Okay, so we've got all the space here. Of course, we want to fill it completely. We don't want to draw over the writing on the question, but we do want to make sure that we are drawing full. We're just going to zoom in a little bit more to see. So you can see we've got different zones here, which are soft, and then other zones which are sort of pebbly, like here. And then we've got this dark, very thin zone on the inside. So those are the zones that we are now aiming to draw. So if we start with that inside, we've got a break here, but then other than that, it's pretty constant all the way around. So if we start there, you would of course be doing this with a lovely sharp pencil. All your drawings must be this is going to be too big, sorry. Using an HB or a B pencil is a good plan, because then of course you can erase it nicely as well. If you use a pencil that is too hard, erasing doesn't work so well. Okay, this is an awful drawing. You can see it is a very different shape. So we'll go one last attempt to get this better in miniature. Okay, so we won't go for quite as much detail as shown. You would, of course, go for all those little wobbles on the inside. One smooth line. Okay, we got that one. So there we said that is a very thin layer on the very inside. Okay. This is much more of a challenge to do with a digital pen than I had anticipated, I must admit. Okay, so now we want the pebbly layer that's running around the inside. So that pebbly layer runs sort of there, then it has a funny scoop in it, then it has a wobble, then it goes and makes a point, then it comes around relatively smoothly. Okay, so all of those loops and wobbles, there's another wobble, and then it goes really, really thin here. So all of those loops, wobbles, thin bits, fat bits, that's what they're looking for, is those level of detail. Okay, so you need to make sure that you've got the detail going in your drawing as well. So now these softer, squashier bits, certainly not squashier in real life, but they look a bit squashy. Okay, those are your cartilage, pieces of cartilage that we're looking at here on the outside. So again, if it stops halfway in a pointy bit, it must stop halfway on your drawing as well. Okay, so then there's a piece that fills in here. Then there's another piece here up to this pointy bit. Then from this pointy bit, we go round again. And then you can see there's a bit of an overlap as well when we get here. So around this pointy bit comes a piece. And all the way around then. So mine is a little bit disappointing, I have to admit. This is not the standard that I would expect on your papers with your pencils. But it gives you an idea of what is going on for this kind of diagram. Now they said we mustn't include any of the other tissues, and they've said we mustn't label. So as soon as we're actually drawn this, now we're finished, and we move to the next question. So the next question, as usual, brings in our magnification. So they tell us that the diameter, let's get it bigger for writing. Diameter of the bronchus in Fig 2.1 is shown by the line AB. So there is the line AB, and it is showing a diameter. Measure the length of AB on Figure 2.1, include the units. So there's a space, length of AB on Figure 2.1, and dot, dot, dot. So we go back to Figure 2.1, and we're going to measure. But what do we always measure in when it comes to biology and it comes to practicals? Okay, unless we're measuring something weird and wonderful and something huge, when we're working with a microscope, we're going to measure in millimeters. So I did take a ruler and I did take a printout and I did actually measure this with a ruler, and I got a length of 55 millimeters. So from that line there at A to the line at B, ruler down, straight across the paper, 55 millimeters. The actual diameter of the bronchus in Figure 2.1 is 1.5 millimeters. Magnification of the bronchus in Figure 2.1 can be calculated using the following equation. Magnification equals length of AB on Figure 2.1 over actual diameter of the bronchus. Yep. Magnification equals image over actual. We're happy with that. Calculate the magnification of the bronchus in Figure 2.1, give your answer to the nearest whole number, and then they give us space for working. One thing we do check, 55 millimeters and 1.5 millimeters, we do have them both in the same units, so we can go ahead with calculating that magnification, because we must always have everything in the same units for magnification. So, magnification equals image 55 over actual 1.5. So that equals 36.7, but they wanted it to the nearest whole number. So that equals 37. Now, what are our units for magnification? Well, there was a hint in my slip-up. We would always say times 37. That is our units for magnification. So I've had the query come through with, is there a handy way of memorizing this formula? Well, missing in action is one of them. You can also do yourself a triangle. And the triangle looks like this. So it's the same like that speed, time, and distance triangle in maths. Okay. So that is your triangle, and then you can see magnification is image over actual, actual is image over magnification, and image is magnification times actual. So there are your sort of options for remembering this one. So, or you can just remember M equals I over A. Generally at IG, then they're going to give you this formula. But in the sort of once off every now and then when they don't give it to you, or in the theory paper, it might not be given to you, then that's your option. Triangle or the one or missing in action. Okay. So, part B. So moving from a bronchus to breathing. A student investigated the effect of exercise on breathing rate. The breathing rates of 5 people were measured at rest and after running for 4 different periods of time. The people rested between each period of running, and the results are shown in table 2.1. So we have running time in minutes, they didn't run at all, or 2, 4, 6, 8, 10 minutes. Breathing rate in breaths per minute, and for each person. So there's 1, 2, 3, 4, 5 people. And there's their average. So 0 running, we had 20, 24, 22, 26, 28. The average was 24. And we can see there's a missing average. So I'm guessing that you can guess what the next question is. Complete table 2.1 by calculating the average breathing rate for 4 minutes of running, space for working. So it's only worth 1 mark. But when they give you space for working and they ask you to calculate something, then it is always worth showing that calculation. So in order to get an average, we're going to sum them all and divide by the count. So we would have 39 plus 41 plus 38 plus 42 plus 40 all over 1, 2, 3, 4, 5. Plug it into the calculator. The answer to that is 40. So then we would definitely go back and put it in the table where it belongs. The average breathing rate in breaths per minute for 4 minutes running time is 40. So then we move to the next question. The student thought that the result for person 2 at 6 minutes, so person 2 at 6 minutes, was an anomaly. Suggest what the student should do to determine if the result is anomalous. See, look at person 2. They go 24, 31, 41, then 52, then down to 50, up to 51. So it's not quite in sequence. We would expect this to be nice and in sequence like person 1, 20, 32, 39, 46, 48, 49. So while the 52 matches person 3 at 6 minutes, it doesn't fall in sequence with person 2. So something funny is going on. So how do we look for an anomaly? Okay, remember, anomaly is something weird, something unusual, something that's different, okay, something that is potentially wrong. Okay, maybe that person by mistake ran for 8 minutes instead of 6 minutes. Maybe there was a problem. How do we address that sort of things? As a repetition. That's what repetition is always about. So if we repeat this 5 times and that person gets 52 every time for 6 minutes, obviously 6 minutes is a problem for them. Okay, so the student should repeat the test for 6 minutes for person 2. And then obviously they can also compare to other people, but it's also going to be one of those things where they're looking at their own. And if we do a comparison, then we can see 52, 52, 51, 46, 46, 46, 44, 44, down to 43. So it seems it's some sort of break in the data for a lot of people from 6 minutes onwards. So the student can also compare to other people. So now we've got 2 things that they can do. Okay, repetition always the first one because that is our best way to deal with weird things going on in our data.